These are your notes for the cell cycle and mitosis. So another word for mitosis is cell division, which is why it says cell division or mitosis. So let's go ahead and get started. All right, so first of all, before we talk about the details, why do cells divide? There are three main reasons why cells divide. The first is if you are a unicellular organism, when you divide, you reproduce, right? So your bacteria do this, because those are all going to be unicellular. Those are your unicellular protista, or sorry, your unicellular prokaryotes. And your protists do it, your protista do it. Those are your unicellular eukaryotes. And actually all bacteria are prokaryotic and all prokaryotic cells are unicellular. If you're a plant, an animal, or a fungus, and you divide, you are going to either grow the body that you're part of, or you're replacing damaged tissue. Those are usually the two ones for the rest of the eukaryotes. So plants, animal, fungi grow, or divide to grow, and divide to repair lost cells or damaged tissue. Right. Oops. The two cells that you get when a cell divides, so let's say here is your original parent cell. I'll label it P for parent. When a cell divides into two, I'm going to draw these smaller because they're going to be about half the size, just because there's two of them. I'll label these D for daughter cells. These are called daughter cells, the ones that are the resulting cells. And then these will eventually grow and they will divide themselves. So this is a cycle that can occur over and over. And because it's a cycle, it can be drawn as a circle. It's just a good visual for it. So let's go ahead and walk through this. There are two main parts of the cell cycle. The first is interphase, and then the second is actually cell division, right? So there are three main parts of interphase, and we'll see this in a second, but I'm going to draw it out here. The first in this kind of light blue area is called G1. This stands for gap 1 or growth 1. Basically, the cell is just gaining cytoplasm and it's building cell membrane because it just divided, so it's a really small cell. It has to get bigger. Then it goes through what is called synthesis or S phase, and you'll see this on the next slide. That's where DNA is going to get replicated. It's going to get copied. And then after that is another G phase. I don't know why it does that, but G2 stands for gap 2 or growth 2. And that's basically just to make sure that the cell is ready for mitosis and there aren't any errors. All the DNA is copied successfully and all that stuff. So all of these together, these three components, G1, S, and G2, are all what's called interphase. They're all part of interphase. Cells will spend about 90% of their time in interphase. The rest of it, the remaining 10% of the time, is dividing, is actually cell division. So the first four sections of cell division are called mitosis, and you'll see that over the next week. This is prophase, metaphase, anaphase, telophase. And then immediately after mitosis, you have cytokinesis, and that's the last step of cell division. So here's basically the same stuff in more detail, right? Interphase was the longest phase of the cell cycle. Remember, we said it was about 90%. That's when the cell is doing its normal job, right? If you're a pancreatic cell, you're making insulin. If you are a liver cell, you're making probably a lot of lipids, you're storing glycogen, and you're probably doing a lot of detoxification, right? So it just depends on what your cell is, but you do your job in interphase, right? Because just by definition, you can't do it when you're dividing. So this is when the cell is working, basically, right? Remember that during S phase, the chromosomes get copied. So during G1, they grow. During S, chromosomes get copied, which is where all the DNA is. And then again, they grow one more time, during G2, right? So those were the three parts of interphase. And here's a summary of those, again, down here as well. So before we actually go ahead and look at some of the images of this, we have to cover some vocab. So a chromosome is just condensed DNA. Condensed DNA. Usually DNA is very, very kind of unwound. It's all kind of spread out all over the place, and it's actually really, really, really hard to see unless it's all condensed. So when a chromosome, when the DNA is all condensed, then you can finally see it. You can actually see the chromosomes. Otherwise, it's really hard to see DNA, and you have to have quite a lot of it before you can actually see it without technology to help. So this is a chromosome. This is a condensed piece of DNA, and we'll talk about a chromatid in a second. But basically, pretty close to the center is a section of the chromosome called a centromere. And that's important. This is called a centromere because the number of centromeres, so the number of centromeres is equal to the number of chromosomes. So here you've got one centromere, therefore you've got one chromosome. And because there's only one kind of piece of DNA here, one chromosome, we're going to call this a single-stranded. This is a single-stranded chromosome. After the cell copies the DNA, right? So after the DNA gets duplicated during S phase, remember S phase is right in the middle of interphase. There's twice as much DNA, but there's still only one centromere. And this is where things can get a little funky, so just be careful. Because there's still only one centromere, there is still only one chromosome. But you're like, well, Mr. Troughton, there's twice as much DNA. Why isn't it 2 chromosomes? Well, remember, the number of chromosomes equals the number of centromeres. There's still only one centromere, so there's still only one chromosome. However, we don't say, oh, it's a single-stranded chromosome. No, no, no. There's twice as much DNA. So to account for that fact, we call it for what it is. It is a double. A double-stranded chromosome. So the amount of DNA has doubled, but the number of chromosomes has stayed the same, right? Twice as much DNA here, but still only one chromosome because there's still only one centromere. And just to clarify, a lot of people think that the chromosomes cross over each other, like the letter X, but they don't. I'm going to highlight one of the one side of the chromosome here. Right? This is half of that chromosome. It doesn't go across. This is the other half of the chromosome. So if you're ever drawing a chromosome, it's very tempting to just draw the letter X. Oops, it did that thing again. Don't draw the letter X. Really, what you should be drawing is kind of 2 little hooked lines like that. Because then you're really showing me which one the chromosomes, or which side of the chromosome is which. Right? So at this point, each piece of DNA, I'm going to label them 1 and 2. So let's say that we started with DNA number 1. Or let's use letters. This is DNA A. This is single-stranded chromosome A. So there's A. Well, A is still there. God, it did it again. I don't know why it does that. And then B is there. So because B is a copy of A, remember we just copied the chromosome to make it a double-stranded chromosome. What we do now is we call these sister chromatids. It would probably make more sense if we called them identical chromatids. But they're called sister chromatids. It does so happen that they are identical, right? So sister chromatids are identical. They have the same DNA because one is literally a copy of the other. So now it kind of makes sense as to why this was called a chromosome with 1 chromatid. right? Its sister hadn't been made yet. So you can call this a chromosome with one chromatid, but usually we call it a single-stranded chromosome. After duplication, it's a double-stranded chromosome with two sister chromatids. The new chromatid, chromatid B, is a copy, an exact copy of chromatid A. All right. These are color-coded blue because they came from one parent. So maybe these came from mom, right? Well, dad has the exact same chromosome. It's might have different genes or different alleles on it, which we'll talk about later. But remember, you get a copy of your chromosomes from mom and you get a copy of your chromosomes from dad. So dad, meanwhile, can be over here on the red side. Dad has his own version of that same chromosome. So here's dad's chromosome A. Again, either a chromosome with one chromatid, or you can go ahead and you can call this just a single-stranded chromosome. Again, there's only one centromere in the middle. Well, after it gets copied during synthesis, during S phase, again, there's still only one centromere. Right, the number of centromeres is equal to the number of chromosomes, so there's still just one chromosome here. But just like with moms, it's now a double-stranded chromosome, right? Each of these is now called, it adopts a new name, a sister chromatid. So there's sister chromatid A, that's the original, and here is its conjoined twin. Here is identical sister chromatid B. And the same vocabulary applies. A and B are called sister chromatids. So let's do some trivia here. Mom's sister chromatid B, so I'm down here, and dad's sister chromatid B. What would you think those are called in relation to each other? Give you a second. All right, here's what they're called. They're called non-sister. These are called non-sister chromatids. You're sister chromatids if you're copies of each other. But if you're not copies of each other, you're just from mom and the others from dad, then you're called non-sister chromatids, right? The last vocab word here is homologous pair. So once you have a double-stranded chromosome from mom and a double-stranded chromosome from dad, and they're the same chromosome, it's just mom's version of it and dad's version of it, the set of both of those double-stranded chromosomes is called a homologous pair. Pair because there's two of them, right? You've got dad's chromosome and mom's chromosome. And homologous, homo means same, because they are the same chromosome. It's just mom's version of it and dad's version of it. So they have the same genes, right? If mom had an eye color gene right over here on chromosome A, dad also has an eye color gene over here on his chromosome A, right? They match each other, they're homologous, right? Remember, we have 46 chromosomes, 23 from mom and 23 from dad. So maybe this is chromosome number 7 from mom. And maybe this is chromosome 7 from dad. Right? They're the same numbered chromosome. It's just mom's version and dad's version. We'll talk more about the vocab too in class. All right. Here we go. Let's talk about mitosis. We're going to go through prophase, metaphase, anaphase, telophase. A good mnemonic for that is pay me any time. PMAT. Prophase, metaphase, anaphase, telophase. So we're right here in the cell cycle, the first part of cell division. So prophase, the DNA is going to condense. It's going to form visual to the naked eye chromosomes, right? The nuclear membrane is going to disappear and the centrioles are going to separate. So here is an animal cell. And here are the centrioles. We're going to go ahead and label some of these components. So remember, the centrioles are unique to animal cells, and they make these spindle fibers. So there's another centriole. These are the spindle fibers. We can go ahead and add that onto our list. Remember, spindle fibers are just cytoskeleton. Here's the nucleus that's getting broken down. Right, the nuclear membrane is in the way of these dividing chromosomes, so it gets broken down. And here are our chromosomes. How many chromosomes are there? Well, there are 4, right? They're all double-stranded, but there are 4 chromosomes. 1, 2, 3, 4. So that's prophase. We've labeled all of these. We have to label the sister chromatids. Well, a sister chromatid is just each half of the chromosome. So let's look at this chromosome right there. Right? That's a sister chromatid, and that's a sister chromatid. Right? So each half of the double-stranded chromosome is a sister chromatid. And then the center points of the chromosomes are the centromeres. Remember, 4 centromeres, 4 chromosomes. In metaphase, think of meta as in middle. The chromosomes, and this is important, the chromosomes line up in the middle of the cell. So all 4 of the chromosomes line up in the middle of the cell, and the spindle fibers attach to the centromere. The spindle fibers attach to the centromere. Specifically the centromere. So I'll say chromosomes. It's specifically the centromere of the chromosome. I don't know why it does that. All right, and everything else is the same. These are the centrioles here, right? These lines are spindle fibers. And then you guys can label the chromosomes in the centromere. Right, chromosomes are there, the double-stranded chromosomes. And then the centromere is right there in the middle. During anaphase, think of anaphase, ana as in apart. Here's what happens. The sister chromatids get pulled apart. Each half of the double-stranded chromosome, the sister chromatids get separated. Right? They get ripped apart. They each keep part of the centromere, though. So how many chromosomes are in the cell at this point? How many chromosomes are in the cell? Well, how many centromeres are there? Right now there's 8 centromeres. Right? Each sister chromatid takes part of the centromere, which means there are now 8, albeit smaller, there are 8 centromeres. Right? They're half the size they used to be. So the number of chromosomes, pardon the siren, the number of chromosomes doubles. You've gone from 4 to 8. Now, granted, your 4 initial chromosomes were double-stranded, and your 8 chromosomes now are single-stranded, so the amount of DNA has stayed the same, but because you've torn it in half, technically the number of chromosomes has doubled. All right, telophase. Telophase is when the cytoplasm starts to pinch in. It doesn't completely pinch in, but it just starts to, right? The other thing that happens is the centrioles start to get broken down. They are no longer needed. And the nuclear membrane starts to reform. That is the end of mitosis. That's the end of mitosis. Now we have to finish cell division. We have to finish cell division. So cytokinesis. Cyto stands for cytoplasm. Kinesis just is the movement of and the splitting of. So we're splitting the cytoplasm in half. Right. In animal cells, this pinch in is just a complete pinch in, and the 2 cells just kind of separate. In a plant cell, though, and this is one difference, a cell plate forms. It's like a baby cell wall. It's not quite a cell wall yet, but it will be. It's called a cell plate. And this is unique to plant cells. All right, and that's it. So let's go ahead and just look at some pictures. This is a plant cell's nucleus and an interphase. Remember, the DNA is not condensed, so you're not going to see chromosomes, but if you stained the DNA with a dye, and the dye in this case I think is like a purpley blue color, it's just going to look like a big massive DNA, right? If you see that, that's interphase. All of these are some aspects of prophase, right? You're starting to see the chromosomes condense, and that's and they're kind of all together still. They're not lined up or anything in the middle of the cell. So it's still prophase, but you can definitely start to see the individual chromosomes at this point. All right. There is kind of like a little transition here where like they're all getting like in line, but they're not quite in line yet. Here's where we're actually in metaphase. So all the chromosomes are here lined up in the middle of the cell. This red stuff is spindle fiber, right? That's all spindle fiber. Here's where the sister chromatids get pulled apart in anaphase. So you can see half of them go one way, half of them go the other way. Again, the red is spindle fiber. And then here's telophase, where you start to reform your nucleus, right? Just a little bit. The chromosomes are all on one side of the cell, and the cytoplasm starts to pinch inward, right? I am so sorry that this is like making those lines like that. I don't know why, right? So that's telophase. Here's the same thing in some other cells. This is, I think this is a fish blastula cell. Anyway, so here's prophase. Again, here they're all lined up in the middle. So that's the chromosomes, are lined up in the middle. So that's metaphase. They're harder to see, but you can see the spindle fibers. Right here are the centrioles. You can see the spindle fibers being made from them. Here's anaphase. Right, the sister chromatids are pulled apart. Spindle fibers pull sister chromatids apart. Yep. And then you start to see, you can kind of see the cell membrane right here. You can see it there, and it's starting to pinch inward. Right, that's telophase. Right, and then once that ends, cytokinesis starts, and then the cell, you can kind of see it right kind of here is where it's most visible. All right, one last little bit here. It's kind of a funny terminology, but when animal cells divide and the cytoplasm starts to pinch in, you can't see it with your naked eye, but if you have the right microscope, you can see it. It's called a cleavage furrow. It's called a cleavage furrow. So to cleave means to break something, and a furrow is like if you furrow your brow, your face kind of looks like that. You look all angry. Right, so it's a cleavage furrow. The membrane is pinching inward and it looks like downturned eyebrows. Right, kind of like that. And you're cleaving it in half. So this is called a cleavage furrow. It's kind of a funny word. It's just the name of the intersection between those two newly forming cells. Only in an animal cell. All right, this is just a little bit more info on how the cell plate forms. Remember, cell plate is for plant, whereas a cleavage furrow is for an animal cell. The cell plate is actually formed by specialized vesicles that fuse together and then matures into a cell wall. So just some extra, info. Quiz yourself here at this point. So, look around these cells, see if you can identify what stages they're in. You can pause, and you can unpause here in a second. All right, I'm assuming you've paused and tried this out on your own. All the ones I'm going to circle look like they're in interphase. So, again, this is probably interphase, that's interphase, that's interphase. This one I'm going to put a question mark by. Interphase, interphase-ish, interphase. Right, the majority of cells are going to be in interphase, 90+% of cells. It looks like this cell down here, I'm going to label P because that looks like prophase. See the difference between these two? Right, this cell right here, you can clearly see the chromosomes, whereas here it's much, much harder to see. So this is probably interphase here, and this one here is probably prophase. Let's see if we can find a metaphase. This one right here looks like it's metaphase, right? The chromosomes are all lined up in the middle. Some of these are not totally clear. I think those are the most obvious ones. Obviously, this one up here is probably either in meta or ana, or like in between the two of them, anaphase. This one over here looks like it's in metaphase, right there. All right, here are some more. This looks like interphase. Here you can clearly see the chromosomes, but they're still kind of in the nucleus, so that's prophase. Similar story here. This looks like the prophase to metaphase transition, right? It's a little bit later than prophase, but it's not quite metaphase yet. This is clearly metaphase. Here we're starting anaphase. Here we're definitely in anaphase. This is telophase, right? This is where the membrane would start to pinch in around the chromosomes. And let's see, this one looks like cytokinesis, right? You start to see that cell membrane get constructed between the two. So that's cytokinesis. Cyto-K. Cytokine. I ran out of space. All right, so those were your notes. I hope this helped, and we will talk about